Hello everybody. Alright, this is going to be the lecture video for chapter 19, Amino Acids and Proteins. So let's get started. So amino acids and proteins. Amino acids are the building blocks of proteins, and proteins are large to small molecules in your body that do a variety of things. They're structural, they're enzymatic, they're involved in signaling, they're involved in the type of facilitated diffusion that we talked about, passive transport in the last, wasn't the last chapter, but at the end of the lipids chapter. And so they serve many, many roles. Now, the way that they ultimately end up, the form they ultimately have, depends on the sequence of the amino acids, and the amino acids are the monomers that go into these polymers. And so this is an example of just a type of protein. So hemoglobin carries oxygen in our bodies. And when something goes wrong with hemoglobin, then we have what are known as the blood illnesses or the blood disorders. So hemophilia, sickle cell anemia, leukemia, all of these are types of illnesses that are related to either blood cells or hemoglobin specifically. So let's just take a look at some general proteins for some classification purposes. So protein molecules can be very, very large. Sometimes it's hard to study them because they have so many different amino acids that make them up. And sometimes there are so many different proteins in a cell that, I mean, not sometimes, all the time, that it's hard to separate them in order to get the particular protein that we're after. But they do have some general classifications. And so while I'm sort of talking about the ones that are dissolved in the cytosol, the insides of your cell, and those are important because a lot of the time those are enzymatic, meaning that they do some sort of chemical reaction. And disease is often related to malfunctioning or something going wrong with those particular types of proteins. But anyways, what's shown here in this cow that you're looking at is its horns. His horns are actually made of protein. Your hair and your fingernails, similarly, are made out of protein. Or different, I think the protein is keratin in our bodies. And so structurally, enzymatically, again, I mentioned a few different types of roles that they play. So a protein is defined as having at least 20 amino acids linked together in a polymer. Their characteristics and function depend on the order of amino acids. So different orders will lead to different functions. There's a neat example. There's a three amino acid protein. It's called a tripeptide. With 3 amino acids, and if you put them in one order in our bodies take that and they do something very specific with it. If you switch the order of those, use the same 3 amino acids, it plays a completely different role in the body. And I don't remember the exact example to share with you at the moment, but that's how specific they are. Even with 3 amino acids, the order that you put them in really changes the activity that they can have. They can form structural components such as hair, I mentioned that, nails, muscles, cartilage. They can be enzymes, and they can also transport. We talked about LDLs, we talked about HDL. These were proteins that were associated with the hydrophobic molecules, the lipids that we were trying to transport, like cholesterol and triglycerides and that sort of thing. Or oxygen, as we talked about just a couple of minutes ago. Oh, I'm sorry. I always do that. Okay, so here are your classifications. They can be structural. They can be contractile. We didn't talk about one of the most obvious examples of proteins, right? Your muscles. Transport. They can also be storage. They can store nutrients. So here's an example. Casein stores the protein in milk. Ferritin stores the iron in the spleen and the liver. So ferritin is the protein that we have that stores iron so that as we make our blood cells, and they all need to have, the hemoglobin anyways needs to have an iron in the center. This is where it can come from so that we don't have to be getting it every day in our food. They can be hormones, they can be enzymes, and they can be for protection, and not in the sense like a shield, or like a hard outer coating. But I guess that's also true. Like I could think of your hair as sort of protecting the top of your head from sun rays, but the example given here is more like your antibodies. So this is a general schematic of an amino acid. So this here is what we're looking at. So an amino acid has a carbon-nitrogen backbone, and we always read the amino acids, or not just the amino acids, but the peptides or the proteins, from the N side. So we start on this side. And every amino acid monomer can be an NCC, and we'll see lots of this repeating NCC, NCC. We always start from what we call the N terminus, which is the side I mentioned, and we always read toward the C terminus. Oh, sorry. We end here. You'll notice that every amino acid, and thus every protein or peptide, is going to have two ends. It's going to have a nitrogen-containing end that at some pHs is going to be charged, and that's the ammonium group. And then a carboxylic acid group. So remember, there's an H that would be here. It's not here at the moment. It's shown missing, but it would be there. At different pHs, it's going to come off, though. In like basic conditions, it'll come off. Okay. We call that the carboxylate group and the ammonium group. Now there's also a side group called the R group, and this is the group that changes depending on which amino acid you're dealing with. So we're looking at a generic amino acid, and the amino acid in this case has a CH3 as the R group. Now this isn't the simplest amino acid, because we could just have an H there. And actually when we look at all the amino acids, you'll see that H will be what we call glycine, and then this is what we'll call alanine, and then as we stick more and more groups on there, they'll get different names. But this is the group that changes. The carbon that has that R group on it is called the alpha carbon. And I think that's about all we need to look at with this general amino acid schematic here. Now, the lab you're doing this week, or you're looking at the videos anyways, is about how we separate amino acids based on their charge. And basically by their charge, they're more polar or they're less polar. If they have lots of charges on them, then they're very polar, and they like water, and they tend to like, in the lab, the paper rather than the solvent. And if the amino acids have really long R groups that have no charges on them, then they tend to like the solvent better, and so they'll climb up with the solvent and not stay on the paper so much. So watch the lab videos, read the lab, and hopefully you can make the connection between how the amino acids are actually moving up the solvent in that video. All right, so at physiological pH, so that is about, for us, somewhere right above 7.18, something like that, 7.2. Both of these groups are ionized, okay? So at really, really low pHs, this O would have an H on it, and so it wouldn't have a charge. At really high pHs, this one's going to also lose its H, and it'll lose its charge. But right in the middle, we have this little situation. We actually call this a Zwitter ion. The amino acids that have a net charge of 0 are called Zwitter ions. And actually, the pH at which this happens, we call the isoelectric point, and we call it the Pi. So Pi equals the pH where net charge is 0. Anyways, here we're just looking at these two charges. We're not focusing on the R group. In fact, this R group has no charges, and so That's why it doesn't matter. We're going to see that there's at least 5 amino acids that have R groups that do have additional charges on them. So as far as classifications go, there's a few different groups. We have a non-polar group, we have a polar group, we have polar neutral, we have polar acidic and polar basic. Excuse me. Need a little drink. Okay. Let's take a look at them. Now, differentiating factors that are going to matter here. The polar basic groups are going to have these NH3s. These polar acidics are going to have carboxylic acid groups on them. Of course, we could just say ammonium or amine. It's going to depend on the pH. And this is carboxylate or carboxylic acid. Again, depending on the pH. Our polar neutrals, these are polar, but they don't have a charge on them. And so these are our OHs, our SHs, and our amides. Okay, and then you'll see there's not a whole lot of a difference between the polar amino and the polar and the polar neutrals, but let's take a look. So I'm going to show you a list of them. We'll jump back to the little 3-letter codes in a second. Okay, so these are our non-polar amino acids. There's 9 of them. The slide I skipped basically is telling you that for each of the names, there's a 3-letter and a single-digit code that we can give them. And so you can see here for glycine, it's GLY and the single-digit G. This makes it easy when we're writing out, tripeptides is even easier, but when we're writing anything out, I'll show you. If I want to write all 9 of these amino acids in a row, which is how you would name really long peptides like systematically, you'd list all their names. It's a lot easier to write GLY, ALA, VAL, right, than to write out the whole thing. It's even easier to go GAV. You can fit a whole lot more on a single page. So these are their codes. So the polar amino acids are next. There's a few of those. So these are polar aminos and neutral R groups. So that was 9 plus these 5, so that's about 14. Wait, there might be missing one. Let's see. 1, 2, 3, 4, 5. Maybe I can't count. Oh, my bad. There's 6 here. So this is all of them. So these are our polars. I mentioned there's not a big difference between just polar and polar neutral. These are all really polar neutrals. They're polar because they have these really, they can hydrogen bond, these guys, really electronegative atoms in them. That's what I wanted to say. These guys are really electronegative. So, and you know from the past chapters on how alcohols and how amines and how those nitrogens and those O's can really, try to pull on each other and make all these little tight connections. That's what makes them really good at interacting with water. Of course, that's what makes them polar. And then sulfur, it's right under oxygen, has the same sort of abilities to hydrogen bond. It's just not as strong, so we don't really technically call it a hydrogen bond. And then our polar charged. So we've got 2 classes. We've got the acidics. These 2 have carboxylate side groups, and at neutral pH or physiological pH, they are charged. In the lab, under really acidic or basic conditions, of course, we can get those to change. Now, that doesn't happen too often in our bodies, but we do have a stomach full of hydrochloric acid that has a pH down near 2, and so amino acids that are in there would definitely not have this negative charge. They'd have H's on them. There'd be an H right there and right there. So aspartate and glutamate come from aspartic acid and glutamic acid. So whenever you hear those other terms, you got to realize those are also amino acids, aspartic acid and glutamic acid. And then our basics, there's 3 of those. These guys all have nitrogens on them, and that have all gained an extra hydrogen. So they've all got those ammonium charges on them. Histidine, lysine, and arginine. We saw in the last chapter that histidine is what we get histamine from. All right. So you want to be able to sort these into their groups to know which ones are polar, which ones are nonpolar, which ones are charged. So let's see, serine. So you should draw it. You don't have to know this for the test off the top of your head, but you should be able to do this. And so I'm going to try to do this off the top of my head. So I know there's an NCC. And I know this is what that side looks like. I've got all my H's here. Circle my charges. I know that there's an hydrogen on my alpha carbon and then an R group. And then serine, I happen to know, contains the SH group in it. And so I think this is my amino acid. The 3-letter is probably SER. We can check that. And the 1-letter abbreviation, I would guess for serine is S. But now I really need to know, so we're going to go take a look. So serine is here. Oh, I was wrong. I was thinking cysteine. And even with cysteine, I was wrong. I left off the CH2. And I was totally off on just the spelling even. That was really, really bad. All right, so for serine, I did have the 3-letter and the single-digit code right, but I mixed up my my OH and my SH and left off a CH2. So let's go back and fix it. So I'd have to come in here, probably erase this guy. All right, now the real thing had a CH2 and an OH. And again, you'll probably draw a couple amino acids on the homework. Beyond that, you're not going to have to draw any amino acids. That's not the point of this class. But you should understand the structure. You should know what's always there. You should know what's variable, right? This is always here. And that this is the R group. Okay? It looks like I didn't even see the second half of that question, so aspartate was on there as well. Cystinuria. Cystinuria, so it has something to do with cysteine. A rare inherited disease, approximately 1 out of 7,000 people. High concentration of cysteine in the urine. So if you have too much cysteine, it looks like it forms into these really nasty looking crystals. Cysteine stones. Yikes. When 2 cysteine molecules form together. Now cysteine is the one I was thinking about, and when the book said serine. Cysteine has the S's, SH's in them. Sorry, these sulfur groups. And these are what are responsible for the curly hair. We did talk about that. Curly hair has a lot of disulfide bonds in them, these cysteines. If you have a lot of cysteines in your hair, you have more disulfide bonds and you've got more curl. When you get your hair chemically straightened, they go in it, they break those disulfide bonds. Now, this is a similar reaction that's happening that's causing these crystals to form. So again, when you have cysteines, you have these SH's. They form something called disulfide bonds. This is important. I'm mentioning it because later in this chapter, we're going to talk about proteins and how they fold together and their tertiary and quaternary structures. And mainly in the tertiary structures, we're going to see cysteines doing this, forming disulfide bridges. So I'm emphasizing it. You should know it. Sorry. Okay, so let's move on. Amino acids can link together to form proteins. So a peptide bond is the bond that actually forms between the amino acids, and linking 2 or more together forms a peptide. So you're seeing here glycines, carboxylate, and alanine's ammonium group are going to link together. We're going to lose a water. We're actually going to lose this O as well. So all of this right here is going to come out as a water. And we're going to have an amide bond. Two or more is a dipeptide. Three or more, a tripeptide. That goes on like a trend. We can call them polypeptides for short. So again, peptide bond is the special name for the amide bond formed between amino acids. This one is another version of what we just saw. Instead of ball and stick models here, we're seeing it more in the expanded view. And again, we should just recognize that we're going to lose a water and then just connect these together. So that's what's happened here. NCC. NCC. We're going to see that in our peptides. This is a good spot to emphasize that when we start to hydrolyze peptide bonds, break them apart, that's where we're going to break them. After the carbonyl carbon, and that nitrogen. As far as naming goes, there's a general trend. I mentioned you always read from the end side. You just say the name of the amino acid with a YL ending all the way to the end, and then you say the full name of the last amino acid. So if we had 3 amino acids, alanine, glycine, serine, it would be alanyl, glycyl, serine. So you're just dropping the ines or the eights. Alanine, alanine, alanyl. Glycine, glycyl. Serine, ceryl, if there was, another one here. Phenylalanine, phenylalanyl. Yeah, that's how it goes. Now, most proteins have too many amino acids to name this way, so they just have common name. Let's see, let's answer this question. So this is phenylalanine. This is cysteine. And this is alanine. That's my guess. It's been a while. N-terminus. It's going to be the side with the N on it. C-terminus. It's going to be this one. So I'm going to say this is phenylalanine. I'm going to say this is cysteine. And that the tripeptide is phenylalanyl cystyl alanine. I'm probably wrong. Let's see. Phenylalanine, alanine, and yeah, I got it. Alanine was over here. Phenylalanine here. Phenylalanyl cystyl alanine. Yikes, that's a tongue twister. Yeah, those are fun. So, of course, there will be a test question that looks kind of like this. You'll have access to all of your amino acid sheet, I imagine, while you take the test. So don't stress on that too much. Polypeptide is 50 or more amino acids with biological activity. And we define primary structure to just be the sequence of amino acids. So this is a really small amino acid protein here, or a small strand of peptide, I should say. This is a tetrapeptide. The primary sequence of which is alanine, leucine, cysteine, methionine. So just the order of amino acids is what primary structure is. It sounds misleading, structure, like it's some kind of building or arrangement. It's just the order. So this thyroid hormone that stimulates the release of thyroxine, tripeptide, glu-his-pro. So this is telling us what I was telling you a little earlier. The specific sequence makes a huge difference. Just changing the order of this, keeping the same 3 amino acids, this would no longer do the job that it does here, releasing this, this thyroxine. So primary structure is the sequence, and the sequence is very specific and matters. Here's insulin. Insulin's small enough that we can show you everything in one go. So insulin is actually made up of 2 chains. So you can see here, it's actually got 2 N termini and 2 C termini. So 2 different protein strands. They're linked together by 2 disulfide bonds. Note that they are between cysteines. So this is how they hold themselves together. There's also an intra-chain cysteine bond. And so obviously this is not the folded final structure. This is just the primary structure. It's just showing you which amino acids are linked and in which order. Okay? All right, draw the structure and give the name for the tripeptide. Glyceryl serial methionine. You can try that. But I would start just by going NCC, NCC, NCC. You already know what's on these ends. The nitrogen is only going to have an H on it. The carbon here is going to be double bonded. All the C's are going to have an H on them, and then they will all get an R group. And that's the only thing that's going to be different. I don't know why I drew this guy down instead of up, but yeah. And then you would go and fill them in. Glycine's easy. There's just an H. Serine, we know that one now. CH2OH. And then methionine, I have to check that one. I don't remember. I think it has an SH in it also. Not an SH. An S. It's close. Okay. Excuse me. Okay, so they're actually having you draw them together and then link them. You could just draw them all in one go like we did. NCC, NCC, NCC. So here's the linking process. And now they're linked. All right, and we already named it. This is how you would name that specific one. Okay, I think that's a little overkill on the naming. But yeah, go for it. This is a nice chance too, also to see how many different ways can we link 3 amino acids. So you can do that. Here's ALA, here's MET, and here's phenylalanine. Link these together as many different ways as you can. It's a fun activity. Okay. As far as essentials go, some of these can be made by our own body, so they're non-essential. The ones that we can't make, we have to get from our diet. These are the essential ones. There's 9. Histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, valine. We get these from our diets. Having a good diet, basically you should have all the vitamins, minerals, proteins, amino acids that you would need. We get them from the proteins. They're not just free floating around in these particular products. These products are rich in that particular amino acid in their proteins, and so that's why we want their proteins. And that kind of says it here. Incomplete proteins from plants don't really give us those amino acids. Okay. Secondary structure. Secondary structure talks about what then happens to that amino acid chain once we let it do its thing. So we're going to talk about the translation process later on in a different chapter on how DNA turns its genetic information into a protein, which is primary sequence, right? Primary structure. But once that primary structure has been determined and those amino acids are linked together, secondary structure is what we call the things that we see. And we tend to see two really common things. One is something called an alpha helix, the other a beta-pleated sheet. And they're shown this way as little, ribbons with arrows or little, like winding around. And so then when you see proteins, you can recognize what's being depicted here. This is an alpha helix. This is an alpha helix, right? These are beta sheets right here. Okay, anyway. Let's take a look. So the alpha helix forms basically between the backbones of the amino acids. It's done in such a way that all the R groups kind of point out. So R groups are pointing out, right? There are no R groups pointing in. And this kind of keeps all of the hydrophobic bits on the inside and hydrophilic stuff on the outside. Now, if these R groups end up being a lot of your non-polar amino acids, then this whole thing is probably on the inside of the protein anyways. But they're all there for a particular reason, to make the protein fold, you'll see later on in tertiary structure, into a very particular way. It's trying to make the amino acid, the protein fold a certain way. But anyways, the hydrogen bonds form between the backbone. So the nitrogen of one amino acid and its hydrogen are finding the carbonyl of an amino acid that's about 4 down the line, and hydrogen bonding with it behind this, right, connecting together. So we see this interaction. Oops, my bad. It gives it a kind of a winding staircase look. So it's called an alpha helix because of the way that it rotates. If you close your right hand with your thumb up, your fingers are curling around to the same direction that this helix is turning. So it's alpha in that sense. Now we do see beta helixes in nature, but they're not as common. This is the more common one. The secondary structure here is a pleated sheet. And there are a few different types of pleated sheets. There's parallel and anti-parallel. We won't worry about that. What that really just refers to are the directions of the chains kind of going in the same way, or are they going opposite? And in this one, these are anti-parallel, because this chain's going this way, this chain's going that way. As far as being read from N to C, there are situations where you can have two parts. Let's see, I don't think I can get it. Yeah, I don't think I got it. So these are both pointing this way, and you can get some. So these would be parallel versus anti-parallel. Anyways, that's going beyond, I think, what I meant to talk about. So the secondary structure beta sheets is just what happens when we get hydrogen bonding that is not happening sort of in the same direction as the chain is growing. That's something else that you might notice from this previous one. Maybe I should erase a lot of that so it's easier to see. So what I'm trying to show you here is the direction of the hydrogen bonding, okay, is actually in the same direction as the alpha helix is kind of going, right, as the chain is growing. That's different than what we see in a beta sheet. In the beta sheet, they are perpendicular to the direction of the chain, whereas the other ones are parallel. So these are to hold chains together rather than to cause the chains to twist. And so, again, this takes on kind of a zigzag shape, and that's what this is trying to show you. I don't know if there's a better picture of it. In both situations, now this is the key. In both situations, secondary structures are held together by hydrogen bonding. Hydrogen bonding. Okay? So if I was to say what holds together primary structure? Primary structure. The peptide bond. That's what holds together the sequence of amino acids. What about secondary structure? Hydrogen bonding. So I was showing you a little earlier, this is what these structures mean now. When you see these little ribbons with arrows and you see these little twists, now you know they're representing alpha helixes and beta sheets. Here's another secondary structure, probably the third secondary structure that is actually worth noting, the triple helix. And this is pretty much characteristic of like collagen. It's several helixes that are wound together into their own helix. So this is a really strong fibrous type protein. Triple helix is what this one was called. Oh, the main point together here being that the chains are held together by hydrogen bonds. So can't stress that enough. Hydrogen bonds, secondary structures. So give this a shot. See if short-term memory is working. So polypeptide chains held together by hydrogen bonds, sheet. Sequence of amino acids, primary. Corkscrew shape, alpha helix. 3 peptides, triple helix. So Alzheimer's disease. We talked about a little bit of Alzheimer's disease relating to some of the neurotransmitters. But the other reason that you get Alzheimer's disease has to do with plaques that form in your head or in your brain. And some of that has to do with this beta amyloid protein. So sometimes this beta amyloid protein ends up aggregating. That means it sticks together. They form these clumps, and that starts to build up. And when that builds up, they think that has something to do with the disease and the onset of the symptoms of the disease. And so it's just worth talking about. This is a 42 amino acid protein. It exists in an alpha helix form. This protein actually, it gets pretty complicated, and I did some of my research at Fresno State actually working in a lab trying to develop drugs that would help with Alzheimer's disease, and they were particularly focused on molecules that would interrupt the formation of these plaques. So it's a heavily researched area at the moment. Let's see. So here's a nice image. So this is showing the beta-amyloid proteins that are normally in this alpha helix form change into this beta sheet form, and in that form they're able to stack together. And so they're trying to inhibit some of the research is working on trying to inhibit this conversion. What it turns out though is it's not that simple. If you inhibit this guy is actually doing a job. The job is not meant to be clumped together, but it's meant to do something else. And if it's not being formed, that leads to other problems. And so it's a complicated picture to try to figure out what to fix and how to change it and kind of cure this disease. Let's talk about tertiary structures. Tertiary structures and quaternary structures are kind of the same. Tertiary structure is the final folded protein. So once you got your primary sequence and you got your secondary structures forming, like your helixes and your sheets, the whole thing needs to fold up into a particular way. And the way that it folds up is determined by several interactions, and we're going to go over those. Quaternary structure is just what we call several tertiary structures that all work together and are held together by some of those interactions. So a good example into a quaternary structure is hemoglobin. So hemoglobin is made up of 4 protein chains, 2 alphas and 2 betas. It's interesting because fetal hemoglobin actually doesn't have beta chains. It has something called gamma chains. And the gamma chains help it bind oxygen at a higher rate than the beta chains would, and so the baby doesn't have to compete as much for the oxygen because it's got like better hemoglobin that could just soak up more oxygen. Anyways, the alpha chains and the beta chains, I'm not sure which is which, they're labeled here as just different colors, are each tertiary structures. So this is a tertiary structure, final folded protein. Here's another tertiary, another tertiary, another tertiary. There just happens to be 4 of them all working together. And we call that the quaternary structure of hemoglobin. And not quaternary because there's 4, quaternary because it's the final structure after the primary, secondary, and tertiary. Okay? There just happens to be 4 of them here. But insulin, we saw, had 2 subunits, 2 chains. Its tertiary structure would still only have, well, actually, that's a bad example because those 2 are linked together, and that's not quite the same example I was going for. If I had to, I would say that in insulin, the first chain and the second chain were their own tertiary structures, and then they interacted together and formed some bonds, and that was their quaternary structure. 2 different proteins working together, all folded up, individually. Let's take a look. So tertiary structure is the final overall folded protein. So here, this one is shown interacting with some kind of molecule. Maybe this is an enzyme of some sort. It happens in such a way that all the parts of the chains that are, or parts of the protein that are similar to each other want to be close to each other. So if these were all hydrophobic parts, that's why they're interacting here. If these 2 bits are supposed to be next to each other, it's because they're hydrophobic. Maybe this guy right here wants to touch this guy right here. Well, then this needs to be something polar, and this needs to be something polar, so they'll find each other. And we'll look at some of those examples. So hydrophobic interactions are the ones that just happen between things that don't like water. So because they're all just trying to get away from water, they find each other. Phenylalanine, nonpolar, leucine, nonpolar, so these things find each other. We tend to find these hydrophobics at the center of proteins. It makes sense too in our lipid bilayer. Remember, we had our lipid bilayer with all the little, what do they call them? The triglycerides in there. They're not triglycerides, sorry, the phosphoglycerides. Anyways, we had protein membranes, or channel proteins that would let ions through, let's say, from one side to the other. These proteins are made up, of a big long chains of amino acids. So all of the amino acids that are right here in the middle of this protein are probably nonpolar, because this whole area right here is nonpolar. All of the amino acids on the outside here are probably polar, because that's where water is. And they're the ones interacting with the water. Hydrophilic interactions. So again, these are probably the ones on the outside of the proteins, especially the ones that are involved in any way, dissolved in the cell or going around in the blood. They have to be polar on the outside or water wouldn't like them. So serine, OH group, hydrogen bonding. So all the polar amino acids, right, not just serine, would be able to do these types of interactions. Salt bridges. These only happen between polar basics and polar acidics. So that's why they're shown here like that. Here's an acidic amino acid, here's a basic amino acid. Negative charge, positive charge. They like each other. Salt bridges is what we call it. Hydrogen bonds. These also form between anything that can hydrogen bond. So carbonyl carbons, nitrogens, this guy can hydrogen bond, anything with an OH on it, anything with an NH on it. Yeah, pretty much. I was going to say, even if we have something with an SH on it, we might still see some O to H hydrogen bonding there. But that still, of course, includes the O, this O. Hydrogen bonds, that's the fourth type, and there's a fifth. Disulfide bonds. This only happens between cysteines. So to sum it up, tertiary structure is a final overall folded protein that includes all of those types of interactions. So the alpha helix, the beta sheet, those are hydrogen bonds only. And then here the final folded protein has disulfide bridges, some hydrophobic interactions, hydrophilic interactions with the water environment, right? We can have hydrophilic interactions within as well. Those aren't shown here. Hydrogen bonding, salt bridges. Anyways, that's what tertiary structure refers to, and those are the forces involved. We could do this. These are both cysteines that contain SHs, so disulfide. This is nonpolar, this is nonpolar. So we would say hydrophobic. So you would have to look up the amino acids if you didn't know. Take a look at their structure, at the very least, classify them as polar and nonpolar, and then you'd be able to answer that question. Okay, quaternary structure is just multiple tertiary structures interacting with each other. And I mentioned hemoglobin, 2 alpha chains, 2 beta chains, and they all work together. Same stabilizing interactions as in tertiary. I think this has a picture of the hemoglobin in the middle. I mean the nitrogen. Sorry, the iron. No, I don't think so. There's a neat difference between hemoglobin and myoglobin. This has a quaternary structure, and this one doesn't. They both transport oxygen. This one does it in the tissue, this one does it through the blood. And again, hemoglobin is 4 chains working together, myoglobin is just 1 chain. You can tell the difference too. Myoglobin is a lot smaller. Similar tertiary structures, it's just that myoglobin doesn't work with other protein chains. So to sum it all up, primary structure is the sequence, secondary structure are those alpha helix and beta sheets, tertiary structure just involves multiple interactions to hold it together, and quaternary structure is multiple tertiary proteins. Sickle cell anemia is one of those weird, it's not that weird, but it's one of those good examples of how just one amino acid change really messes everything up. So in your, I think it's your alpha chain, no, the beta chain, the 6th amino acid down is supposed to be a glutamate, and instead it gets turned into a valine through a mutation. So if you get valine, all of a sudden one of these crucial interactions that would have been there is gone. This was polar, acidic, probably involved in some sort of salt bridge. Now it's nonpolar, so it's not going to salt bridge. And right there, the protein sort of doesn't fold properly. So instead, it gets this crescent shape and then they sort of stack together. Sometimes they even cause the whole shape of the blood cell to be misshapen, and then they break easier. So it's a pretty bad disease. As long as you have at least some normal blood cells, you can get by. But if you're one of those people who ends up inheriting two versions of this bad gene, then you don't have even, half of your blood that's working, and that's when it gets really, really bad. Let's see. This is kind of showing what would happen at like the diagram scale. So these are sickle cell anemia blood cells or hemoglobins, and then they form these little strands that can form these big fibers, and then those can clog your capillaries, cause inflammation, organ damage, and obviously not be doing their job of carrying oxygen to the proper places. Okay, let's talk about proteins. Proteins can be hydrolyzed and they can be denatured. So let's talk about what both of those are. Hydrolysis is just undoing the peptide bond. And so we're going to put water in, right? Hydro. Lysis meaning cut. Water in and cut those bonds. Just like our polysaccharides could be hydrolyzed into disaccharides and then monosaccharides, our proteins can be, our peptides can be hydrolyzed into amino acids. As far as denaturation, this is how we disrupt the forces that are holding together our folded protein. If we properly denature a protein, it will go all the way back to its primary structure. If you remove the agent that was causing that, it should fold right back up again. That's not always true. For example, when you denature the protein in eggs, you can't ever get that egg to go back to how it was supposed to, or how it would have looked. And so some denaturing is permanent. So we'll go over the different things we can use, different denaturation agents. So first, hydrolysis. Now this is important for a few reasons, right? We want to eat protein and get the amino acids out of them, so we need to be able to break them up. So our bodies do this for us. There's a few different ways. We can acid catalyze, so this happens primarily in the stomach. And we also have some peptidases. These are enzymes that go in and break up peptide bonds, like trypsin and chymotrypsin. These are sometimes secreted in the stomach or into the small intestine, and they go and they start breaking up at specific locations the proteins. So it breaks the primary structure by breaking the covalent bond. So here is a peptide, a tripeptide, NCC, NCC, NCC cut, NCC cut, right? Water gets put back in, by the way. So on this C, we're going to get our OH, and then on this N, we're going to get our H. Now, because these are non-acidic, well, physiological pH, this guy is going to lose its H. It won't have its H. It will have a charge. And because this guy, so now the free-floating nitrogen will have a lone pair and there's hydrogens around, it'll gain that H. So this is one of those times where we're going to add water, but we're going to add only O to the carbon, and we're going to add the two H's to the nitrogen. So this only happens because of the fact that this is acidic and this is basic, the nitrogen. And yeah, so that's where the water went, the O and two H's. Okay, these are the things that can denature a protein. Increasing the temperature, so heat. Changing the pH. If we change the pH, those salt bridges go away, right? If I've got this this happening, and then I change the pH to be very, very basic, then this guy loses its H, it loses its charge. These are no longer going to be interacting with each other. So that's how pH is affected. Metal ions. Metal ions will go in there. Metal ions often have really charges on them, really big charges. They're going to go in there and disrupt salt bridges. This guy's going to go in there and just bond to this O. And if that happens, again, there's not going to be an interaction now between this nitrogen and that O. And then mechanical agitation. This is just shaking them around, busting them around, stirring them really vigorously. That's going to disrupt those bonds. When this happens, globular proteins will unfold, and if they're enzymatic, then they no longer do the job that they were doing. So heat causes the hydrogen bonds and the hydrophobic interactions to stop being effective. If you're worried about if that makes it less nutritional, it doesn't. It does make it easier to eat. This is why we autoclave instruments, so if any bacteria or any kind of microorganisms are on there, just high heat causes their enzymes and usually their whole bodies to sort of just kind of melt, but it at least kills them. Changing the pH, I mentioned, will also break the hydrogen bonds, but it mainly disrupts the ionic bonds and the salt bridges. Often we use ethanol and isopropyl alcohol as a disinfectant. This is because they will hydrogen bond with the bacteria instead of the bacteria hydrogen bonding to water. And this messes up just everything that's going on inside the bacteria. So it's another way of just sort of killing it. Alcohol also readily passes through the cell walls of the bacteria, and so that's why it's effective. Heavy metals, silver, mercury, lead, they form bonds with those ions, or they react with disulfide bonds. And so if you have a lot of disulfide bonds, a lot of cysteines, like the insulin, for example, had 3 disulfide bonds that were in its final structure. This could basically knock that protein out. Babies that are born that might possibly have gonorrhea, they can put silver nitrate in their eyes because that will go and kill that gonorrhea. And if you've ever made whipped cream or beat egg whites, then you've used mechanical agitation to denature a protein. I think that's about it. So review the denaturing methods: heat, acid, base, or heat, pH, organic compounds. Did we say anything about organic compounds? Why doesn't that sound familiar? Oh, alcohol and ethanol, disrupting hydrogen bonding, essentially. Heavy metals and agitation. So know about denaturing, and then let's see. Tannic acid is used to form a scab on a burn. An egg white is hard-boiled by placing it in boiling water. What is similar about these two events? Interesting. They're probably both denaturing a protein of some sort. Yeah, don't worry about a question like that. Acid and heat cause denaturation of a protein. Okay, so I guess that's the end of the chapter. So amino acids, they're categorized polar, nonpolar, polar acidic, polar basic. Know those. Know about a peptide bond. Be aware that these groups are pH sensitive and they can change at low pHs. We have OH and NH3. At physiological pH, we have O- and NH3, so they're opposites. We have the Zwitter ion, no charge. And then at high pH, we're only going to have negative charges. Know about primary secondary, tertiary structures, and how quaternary structure is similar to tertiary. And then as far as peptide bonds go, hydrolysis. Amino acids are the product of the hydrolysis of polypeptides. Denaturation happens to the secondary, tertiary, and quaternary structures, and know the various ways that these guys can disrupt those. So I want to test if I say you have a protein and you add a lot of heat to it, in what way, what's going to be most affected in this structure. You're going to say hydrogen bonds, right? If we go and we say you have we add some acid and base, change the pH around, what's going to be most affected? You could say hydrogen bonds, salt bridges, ionic interactions. So know that kind of detail in terms of what you think would be the result of that particular type of denaturizing agent. Alright guys, well I'll post this video, hopefully, well of course you're watching it if I've posted it. I'll open a discussion board so you guys can post questions if you have them. Stay up on the reading, and expect a quiz at the end of the week. Alright guys.